Thanks for the introduction. It is nice to be here and talking to you all. I am Anita Tiwari, a Senior Pharmaceutical Quality Assessor in Delapi ONDP. The title of my talk is ICH Q11 Q&A, a supporting document for the selection and justification of starting materials. In this presentation, I am going to cover brief background of ICH Q11 and its general principles and key concepts, clarifications and recommendations on some critical questions and answers, case study, and lastly, conclusions. ICH Q11 became effective in May 2012. Section 5 belongs to selection of starting materials and source materials. However, Section 5 in this guidance was broad and vague. Industry wanted more clarifications. So, a supporting document, ICH Q11 Q&A, was developed and it became effective in August 2017. As you can see, ICH Q11 Section 5 contains two sections and subsections. So, what is the ICH Q11 Q&A document? It contains 16 questions and answers which provide more clarifications. It clarifies the ICH Q11 general principles concept for the selection of starting materials, and it should be used in conjunction with the ICH Q11 document. It recommends the starting materials selection should be based on the knowledge of the proposed commercial manufacturing process and controls. It also contains a decision tree to clarify Q&A with the support of all ICH Q11 general principles for the selection and justification of starting material. Scope of Q&A follows ICH Q11 and applies to commercial drug substance manufacturing processes submitted as part of marketing authorization applications and/or drug master files. It is not intended to be applied to the manufacturing process previously reviewed unless significant changes are made to the process. Since manufacturing process in each application is evaluated on its own merit, a starting material accepted for one manufacturer's process may not be considered acceptable for a different manufacturer's process depending on their process and justification. This slide shows the summary of general principles described in ICH Q11. Please note that the drug substance manufacturer should consider all these general principles, not just some of them, for starting material selection. I will be covering them in the later slides. However, I just want to emphasize one point here. To distinguish between reagent, solvent, catalyst, or other raw material, a material is considered a starting material if it is incorporated as a significant structural fragment into the structure of the drug substance. However, this should not be the sole basis for the selection of a starting material. For example, late-stage intermediates containing the significant structural fragment should not be considered starting material. This slide shows the common issues we encounter in the submission regarding starting material selection and justification. You can read these issues and address them to improve your application. Though I want to emphasize the last point here. If the drug substance manufacturer is referencing a secondary DMF for a late stage intermediate, they should refer this intermediate as an intermediate, not as a starting material in the application. Now I am going to talk about some Q&As which are more critical and often interpreted incorrectly. Q&A 5.6 clarifies the difference between custom synthesized chemicals, and commercially available chemicals. If a chemical is made in-house or externally, specifically to the drug substance manufacturer's requirement, or available for purchase where the only use is for pharmaceutical manufacture, then it comes under custom synthesized chemicals. If a chemical that is sold as commodity in pre-existing non-pharmaceutical market, then it comes under commercially available chemicals. Please note that custom synthesized chemicals should be justified for each proposed starting material per ICH Q11 general principles, while commercially available chemicals as a starting material generally do not require justification. Regarding Q&A 5.7, a general principle, that is, the steps that impact the impurity profile of the drug substance should normally be included in section S.2.2. For that, understanding of the process and the origin, fate, and purge of impurities are necessary before defining the starting material. Now the question is, what are the levels of impurities that impact the drug substance? If the impurities are non-mutagenic, the level above ICH Q3A identification threshold impacts the drug substance depending on the MTD. For mutagenic impurities, the level above 30% of TTC based on MTD impacts the drug substance. When the drug substance is itself mutagenic, impurities are not considered to impact the impurity profile of the drug substance unless they are above the ICH Q3A identification threshold. Please note that the general principle described in the previous slide of Q&A 5.7 does not necessarily apply when impurities originate early in the process, such as chiral impurities, regioisomers, and persist across multiple steps to the drug substance. In the following slides taken from ICH website, example 4 from ICH Q11 has been expanded to show the different scenarios of impurities that impact the impurity profile of the drug substance. In this example, step 1 results in the formation of the opposite enantiomer of compound B. This impurity persists and referred to as impurity 1 in subsequent slides. Other significant impurities arise in steps 4, 5, and 6. Impurities 1, 2, and 3 impact the impurity profile of the drug substance. Impurities originate in step 2 and 3 do not impact the impurity profile of the drug substance. Therefore, D is proposed as a starting material in this case. In this example, impurity 1, 2, 3 impact the impurity profile of the drug substance. Impurity 2 and 3 originate in step 3 and 5 respectively. Therefore, in this case, D is no longer suitable as a starting material and C should be redefined as a starting material. In this example, impurity 1, 2, and 3 impact the impurity profile of the drug substance and they originate in step 1, 2, and 3 respectively. So in this case, C is no longer suitable as a starting material and A should be redefined as a starting material. This slide shows how to apply ICH M7 principles in the selection of starting material. One should identify mutagenic materials that are likely to be formed or introduced in the manufacturing process. Use hazard assessment elements from ICH M7 Section 6 to determine which of the actual and potential impurities are considered to be mutagenic. Hazard assessment involves an initial analysis of actual and potential impurities by conducting database and literature searches for carcinogenicity and bacterial mutagenicity data in order to classify them as class 1, 2, or 5 per ICH M7 Table 1. If data for such classification is not available, an assessment of structure-activity relationships that focuses on bacterial mutagenicity predictions should be performed for class 3, 4, or 5. A computational toxicology assessment should also be performed using QSAR methodologies that predict the outcome of a bacterial mutagenicity assay per ICH M7. This assessment should be included in the application. The impact level of mutagenic impurities is more than 30% of TTC. Therefore, approaches described in Section 8 of ICH M7 can be used to control mutagenic impurities. Impurity hazard assessment per ICH M7 and application of QSAR are covered by a different talk that I recommend you should listen tomorrow. Q&A document recommends the following approaches for mutagenic impurities. The drug substance manufacturer should consider them before starting material selection. Actual impurities should be assessed for mutagenicity. Reagents and intermediates used in the synthesis from commercially available chemicals to the API should be assessed for mutagenicity, and this should also include upstream steps of the proposed starting material. Mutagenic impurities in commercially available chemicals or synthetic intermediates or formed by side reactions during the synthesis should be assessed by using risk-based reasoning to determine which steps to include in the hazard assessment. A discussion of the risk assessment should be included in the application. If the drug substance is itself genotoxic, the impurities can be controlled per ICH Q3A. The ICH Q11 exception for impurities that persist is also applicable to mutagenic impurities. Regarding general principle, in ICH Q11 5.1.1 listed here, Q&A 5.11 provides more clarification, and the drug substance manufacturer should apply these recommendations for starting material selection. I am going to cover them in the later slides. However, it is important to note that the manufacturer should consider the potential risks from future changes to synthetic route or supplier of starting material during evaluation. Please also note that synthetic route or manufacturing process of the starting material should be included from each supplier in the application. Case study slides taken from ICH website for better understanding. This study applies all the general principles of ICH Q11 together with Q&A for the selection of starting material. Please note that this study is not intended to convey whether or not a given number of steps are enough in the drug substance manufacturing process. As you can see in this synthetic scheme, the manufacturing process of drug substance C9 contains 6 steps, 2 9-isolated intermediates C3 and C5, 3 isolated intermediates C4, C6 and C7, and starts with 2 starting materials C1 and C2. Please note that cGMP pathway starts from the first use of starting material. C8 is also proposed as a starting material. This slide shows the hypothetical drug substance specification. Justification is provided by the applicant for their controls. The impurities that impact the drug substance should be specified in the drug substance specification. Please note that impurities above IT level or above 30% of TTC impact the drug substance. As you can see, two mutagenic impurities C6 and C8 controlled at TTC level based on MDD. C3, C4 and C5 are controlled at acceptable limits in intermediate C7. Therefore, they do not impact the drug substance and they are not specified in the drug substance specification. I'm going to cover these impurities individually and talk about whether the intermediates are suitable as starting materials or not in the later slides. Impurity C9D1 is a distereomer and it is introduced in step 4 during cyclization. It is found above the identification threshold, therefore considered to impact drug substance impurity profile. It is controlled in the drug substance specification. C6 is a mutagenic impurity and found above 30% of TTC, therefore considered to impact the drug substance impurity profile. It is controlled at TTC level in the drug substance specification. Benzyl bromide C8 is introduced in the last step of synthesis. It is acceptable as a starting material because it is commercially available commodity in non-pharmaceutical markets and contains significant structural fragment into the structure of the drug substance. Therefore, it is not a reagent. Impurities in benzyl bromide do not impact the impurity profile of the drug substance as they are controlled in the starting material specification. However, if benzyl bromide did contain impurities which needed to be removed prior to its use by API manufacturer, then purification steps should be included in section S.2.2 and performed under CGMP. The specifications for pre-purified incoming material and purified material should be included in the application, and purified material should be considered as a starting material. C3, C5 are non-isolated intermediates, therefore they are not suitable as starting materials. C7 is the last intermediate. It steps upstream, generate impurities which impact the impurity profile of the drug substance, and there are not enough manufacturing steps under CGMP. Therefore, C7 is not acceptable as a starting material. C6 is an intermediate step upstream, generate impurities that impact the impurity profile of the drug substance. Step 4 needs to be controlled to ensure the required stereoselectivity, and there are not enough manufacturing steps under CGMP. Therefore, C6 is not acceptable as a starting material. C4 is an intermediate and it is formed early in the process. Steps upstream are simple and the impurities originate early in the process do not impact the drug substance. The enantiomer control in C2 specification, spiking studies show that it is well below the IT level in the drug substance. Please note that if the enantiomer of this material did carry over to the drug substance at an impactful level, it could be considered an impurity that persists across multiple steps. Radioisomers can be controlled in C1 and or C4 specifications, and there are multiple chemical transformation steps are described in section S.2.2. Therefore, C4 can be acceptable as a starting material, provided it was justified in accordance with the other ICH Q11 principles. So, the conclusions are: proposed starting materials should be justified based on all general principles outlined in ICH Q11 Section 5. Designation of starting materials should be based on process knowledge of the proposed commercial process. Understanding of impurities, whether non-mutagenic, mutagenic, or persist, generated or introduced in the upstream or downstream of the process that impact the drug substance, is necessary before selecting the starting materials, and the steps which impact the impurity profile of the drug substance should be included in section S.2.2. Hazard assessment of mutagenic impurities even present in the starting material manufacturing process should be performed per ICH 7 and included in the application. If additional purification steps are performed for commercially available starting material, these steps should be included in section S.2.2 per ICH Q11 Q&A 5.14. Thank you very much for your attention. This wraps up my presentation. If you have any questions, please type them into the Q&A box in the bottom right hand corner of your screen so that they can be addressed during the panel Q&A after this session. Please also follow this slide regarding additional questions, submission, and cross-referenced posters and presentations. Thanks again and have a nice day.